Hello everyone, I Assistant Professor V Shete from Department of Microbiology, KG Somaya College. In the last online lecture, we have started the study of action of the antimicrobial agents on the bacteria. So in last lecture, we have studied the cell wall inhibitors. In this online lecture, we will study the cell membrane inhibitor. Right, so let's start. What are the cell membrane inhibitors? These are the antimicrobial agents, right? The antibiotics, the drugs, which will do its action or which will do its work by inhibiting the synthesis of the cell membrane. Now you can see in the previous slides, we have saw the structure of this bacteria. In that we have observed the cell membrane. Where is the cell membrane present? It is present just beneath the cell wall, right? So now cell membrane, what is the role of the cell membrane in the bacteria, that we will study. Okay, so first, it is present just beneath the cell wall. It is the most vital organelle of the cell. Now cell membrane or the plasma membrane is a lipid bilayer which is the semi-permeable. Now focus on the word semi-permeable, which means it will allow the nutrient transport inside and the outside of the cell. It will allow the nutrients from outside that are important or that are essential for the bacterial growth development, and also it will responsible for excrete some toxins from the bacterial cells which are harmful to the bacteria, so they will exit that toxins through this membrane. Okay, then it is responsible for proton gradient formation, that is the energy metabolism. Now as they will responsible for transferring certain molecules, chemicals or the nutrients, macromolecule, micromolecules, so they will produce the proton gradient due to the charges, right? And therefore it is responsible for producing the energy metabolism, right? Then they can also have the proteins attached on them which will help the bacteria for the cell to cell contact or any reactions, the proteins, right? Or to synthesize some enzymes, right? So therefore the cell membrane, we can see it is a very important part of the bacteria. And the most important function of the cell membrane is what? To allow the certain specific nutrients required for bacterial cell growth and to excrete certain toxic materials from the bacterial cells. All right. Therefore, a little scratch if made in the cell membrane, then the bacteria will lyse. Okay, that will kill the cell. Now we will study the antimicrobial agent that are doing this job. That is, they are synthesizing, they are inhibiting the synthesis of cell membrane or the plasma membrane. Now first we will study the polymixins. Okay. Now the polymixins are extracted or are produced from the Bacillus polymixa. Okay. Now what are they doing? They adversely affect on the normal permeability characteristic of a cell membrane. Okay, and this type of the drug, the polymixins, are particularly effective against the gram-negative organisms. This polymyxin will come under the bactericidal drugs that they will kill the cells. Okay. Then they are mostly used in the ointment, that means the applications. They are mostly used in the way from the ointment or in the wound or as a bladder washout. Okay, they can used in such modes. Then we will study its main mode of action. What is its mode of action? How it is doing the cell membrane inhibition. So now the cell membrane is the most essential part of the bacterial cell that we studied, right? Because it is involved in the proton gradient formation, that is the energy metabolism, right? Then the polymyxin have a smaller polypeptide ring which is attached to the polypeptide chain terminating with the branch number 8 or 9. Okay, now this ring will have 5 free amino acid. This ring will have free amino groups associated with the diaminobutyric acid, that is the DAB, diaminobutyric acid. Okay, and that is why it is carries the 5 positive charges, right? Because it is having the 5 free amino ring. In the structure of the polymyxin, you can see there are the 5 free amino acid groups are present. Therefore they will have the 5 positive charges which are free to attached, and therefore we can call it as the hydrophilic ring. Okay, now along with the hydrophilic, the same molecule will also have the hydrophobic side chain in the opposite end. That means they will have the both molecule, hydrophilic as well as hydrophobic. Okay, now the positively charged ring or the amino group will bind to the, they will act like an cationic detergent. Cations means positively charged, right? So they will act as the cationically charged detergent. They will interact with the LPS and the phospholipids in the outer membrane, that is the lipopolysaccharide and the phospholipid, okay, which are the essential parts for in the cell membrane synthesis. Okay, now they will bind the electrostatically the phosphohead. The phosphohead or the phospholipid is the negatively charged. Therefore the positively charged amino group will bind to the negatively charged phosphohead of the phospholipid. Okay, and due to this, what it will do, it will replaces the Mg+2 ions, magnesium ions. This normally magnesium ions will contribute to the membrane stability. Okay, but now magnesium ion will not bind. If you know the structure or the charges of the magnesium, it is the Mg+2, right? So basically, if our antibiotic is not present in the process, say for instance, so naturally the Mg+2 ions will bind to that phospholipid negatively charged, right? Negatively charged phosphohead of that phospholipid. That is responsible for membrane stability. It will do transport of the magnesium ion and that will stabilize the membrane. Okay, but now our antibiotic is present, the polymyxin is present, which will bind, and our polymyxin is also having the positively charged 5 free amino acid groups that will bind to the negatively charged phosphohead group of the phospholipid membrane, phospholipid and the LPS, which is present in the cell membrane. Due to this, it will replace the magnesium ion with itself. Okay, and therefore in the outer membrane, and therefore it will increase this cell permeability. Okay, now the same time, that means it is binding to the negatively charged phosphohead, right? And at the same time the fatty acid, that is the LPS chain, inserted into the hydrophobic inner region of the membrane. Okay, now as I mentioned, it will have both the end, hydrophilic and hydrophobic. So the hydrophobic chain, okay, hydrophobic chain, what it is doing, the fatty acid chain inserted into the hydrophobic region of the membrane, and due to which it will alter the cell permeability, right? It will alter the cell permeability first by inhibiting the attachment of the magnesium ions, which are responsible for cell stability, and second, it will insert the fatty acid chain into the hydrophobic end region, okay, and therefore it will alter the cell permeability. Now as the cell permeability will be altered, which means what, it will alter the transport of the nutrients from inside and outside the cell, and therefore the proton gradient, that is the energy metabolism that is producing due to this nutrient exchange, right, it will give energy to the cell. Now the proton gradient is disturbed, right, therefore the ions transportation will be get disturbed, and therefore the molecules will be lost from the cells, that is the essential molecules that are present, okay, that will get lost from the cell, and therefore then cell will not able to do its functions, cell will lysed, cell will kill, cell will get killed. This is the mode of action of the polymixin. Polymixins are cell membrane inhibitors. Polymixins has 5 free positively charged amino groups plus, which is hydrophilic. Also it will have the hydrophobic chain. Now what it is doing, it is binding to the negatively charged phosphohead of the phospholipids. Okay. Now what this result? This will result to inhibit the attachment of the magnesium ions to this negatively charged phosphohead, okay, plus simultaneously it will insert the fatty acid chain that is present in the cell membrane into its hydrophobic end, and therefore these both processes together will alter the cell membrane permeability. Now the cell membrane permeability is getting disturbed, the proton gradient formation will disturb, right, due to which ion transport will be disturbed or will be get harm. Therefore the essential molecules will get lost from the cell membrane, because cell membrane is permeable, right, so the essential ones will also get lost from the cells, and therefore the cell will get killed, it will get lysed, since it is not containing any essential nutrient that is required for its growth or its role, its development, so it will not able to do the function only, right, so it will get killed. This is the role of the polymixin. Now second we will see, that is the monensin. Okay, in the monensin it is called as the ionophore. What is the ionophore? A substance which is able to transport the ions through the lipid layers. It is called as the ionophore. Lipid layers are present where? In the cell membrane, right? Lipids are the component, the phospholipids and the LPS are components of the cell membrane. Now this monensin are the ionophore, that means it will able to transport the ions through the lipid layers. Now it will changes the pH and the sodium potassium balance in the cell. Now the potassium and the sodiums are very important nutrients which will maintain the pH of the cell and which will maintain stability and which will balance the nutrient transport in the cells. Okay. Now, as this changes in the pH and the sodium potassium balance, it will lead to the critical disturbances in the cellular processes in the cellular membrane synthesis, and which results into the cell death. Now, monensin and some of its derivatives have shown activity against the gram-positive bacteria, including the micrococcus, then bacillus and staphylococcus. Okay, then monensin can transport sodium ions through the membrane in both the electrogenic way and electroneutral manner. Electroneutral means what? There is no charge, which means either it will remove all the potassium and it will remove all the sodium from the cell, or it will produce very excess of the sodium, that is the electrogenic. Okay, and the electroneutral means there will be no nutrient, there will be no the potassium, and therefore in both the ways it will transport the sodium and it will inhibit, okay, it will block the protein transport. And therefore the sodium potassium balance will be disturbed, right? The pH will be disturbed. Now these are the most optimum conditions for the cell stability and the cell growth, the pH and the potassium sodium balance. Now these both crucial things are getting disturbed, therefore the cellular processes will not take place properly. It will block the intracellular protein transport, okay, and exhibit antibiotic, anti-malarial, other biological activities. Okay, it is also having the anti-malarial activity against the malaria it can use. All right, this is the mode of the monensin antibiotic antimicrobial agent, which is coming under the cell wall inhibitor. In the right hand side you can see the structure of the monensin. It is basically a ionophore. Ionophore is what? A substance which transports the ions through the lipid layer. Okay, it will make changes in the pH and it will disturb the potassium sodium stability in the cell. This is the monensin. Now we will see the second protein synthesis inhibitor. In the cell membrane inhibitors, we have studied two antimicrobial agents. First one was the polymixin, right? Polymixins, and the second is the monensin. Polymixin has free positive charge amino groups. Monensin is the ionophore, okay, which is responsible for changing the or disrupting the potassium sodium balance and the pH. I hope you understood this online lecture. If you have any doubt regarding this, please ask in the comment section. Thank you for watching.